Hi everyone. Thank you. Thanks to the audience for joining us. We have now nearly 100 people who registered to be live with us today. Before I start to say anything, I would like to address my deep, sincere condolence to the Israeli people for what they suffer now. It's very sad, horrific, and we are all with you. So that's what I wanted to say just before to start. Today I have a fantastic guest, Dr. Ari Gargir. He's the founder CEO of RedC Biotech, great company that is developing maybe one of the most precious solution to one of the most critical problem in the health issue in the worldwide. So thank you very much, Dr. Gargir, to be my guest today, and I'm very happy to have you today because of the circumstance. Of course, it's very important to have you today.
Thank you very much, and I'm happy to be here and present RedC. Good afternoon to or morning to everybody, and I'll be happy to tell you and about RedC. We're doing something that today more than ever is important, and emphasized that it's needed, and I hope that we'll be able to contribute with this a lot.
Perfect. Maybe Dr. Gargir, could you maybe directly talk about the problem you address before even we start by discussing about your background and who you are? Maybe we need to bring to the mind of our audience how the problem you address is critical, and maybe you can start with that.
So let's for the very top level. So RedC Biotech is developing an industrial process that will enable the production of unlimited red blood cells for mostly for life-saving transfusions and for advanced therapies. Um, it's a revolutionary technology. It's a revolutionary solution that will help the blood supply all around the world. It's an immense market. The potential of the market is very big at this point, and I'll perhaps explain about this later on. But we're still at the, maybe half of the amount of blood that's needed for the world. And so the actual potential of what we're doing is much larger. And it's, of course, a humanitarian, it has global impact. And what we're doing will have impact on countries all over the world, advanced countries and low-income countries. So this is what we're doing. And the need, perhaps you would like to go about the actual need and analyzing the need perhaps later, and we'll go about that all of the aspects of it.
Sure. I just wanted to highlight how much the shortage of blood transfusion is a very critical issue in the health for the health of the human being worldwide. It's not just something that affect let's say developing countries. So it's also touching developed country. Do we have some numbers to share? How many people are affected? How many people die because of blood transfusion shortage and or have a bad consequence of it?
So around the world, there are about 120 million blood units that are donated every year. 60% of them are in the high-income countries, meaning that maybe a quarter of humanity is enjoying two-thirds of the blood that is donated. So it is calculated that there are about 100 million blood units that are in shortage every year. This is contrast between high-income countries and low-income countries. On the other hand, also in high-income countries, there are occasional shortages. Blood, blood is given. The only way people can get blood is from donation. There's no other way today to supply blood to human beings. And because of that, excuse me, and due to that, the site is at the whim of the goodness of people. There are holidays, there's bad weather, there are pandemics, there are emergency states, and also in places that have an actually very good blood supply, sometimes fall into shortage. The 19 pandemic showed it. Many countries, almost all of the blood transfusion agencies, were in severe supply challenges throughout the pandemic. And I think there's an understanding today that there needs to be an alternative to these resources. We cannot only depend on the altruism of people, even though mostly people are very happy to donate and they feel good about saving lives.
Absolutely. So there is the first problematic is the donation. There is also the of the blood. It's a living, or it's the cells are living cells, so they can't be stored forever. They can't be freezed. They have tried in the past, to many a protocol to freeze them and to unfreeze them, but there always a lot of loss. The cell die, and at 2 degree, the top of your fridge, the has a limitation. We can't storage blood forever, so it's a can we say it's a lean manufacturing process? It's a
yeah. First of all, red blood cells have a shelf life, and them are stored at 2 to 8 degrees Celsius, and at that temperature they can last for about a month. So the shelf life is limited, but it's also known that their quality starts diminishing after a couple of weeks. So they're not exactly as good as they were when they were donated. There are additional problems or challenges that the blood donation industry have with the blood supply. One of them is the fact that each unit is a batch of its own. And so each blood unit has to be tested for 7 to 8 antigen diseases, contaminations like HIV, hepatitis, and each one of these blood transfusions has to be typed, because as you know, there are the different blood types. There's the ABO groups, which O is the one that could fit everybody. There's the RHD group, which in the minus, the negative, fits most people. But the distribution of the blood types distribution of the donations does not exactly fit the distribution of the needs. For example, O negative blood type is considered universal. You can transfuse O negative to every man and woman, generally without testing, if an emergency. When a person would go into an operation, there are additional tests that are done in order to make sure that you have the exact blood type. That's called cross-matching. But in emergency, you could give O negative to almost every human, especially O negative is most likely given to women at the fertility age. Maybe we won't go into the exact reasons to that. O positive can be given to everybody, to all the rest, all older women or men, without testing. But the distribution is 6% O negative, when you would like to give that to almost 50% of the population, and 30% O positive when you want to give it to the others. So there's always a problem in the stocks, in the inventories. It's very difficult. The shelf life makes it difficult, the logistics, and of course running after the people, the contacting and bringing the donors to actually give the blood is a problem that is actually growing as humanity goes forward. People are less likely to donate in these days. The population is growing. The population of elderly people is growing. The population of cancer patients is growing, and about 20 to 25% of the blood donations go to oncology patients. So a lot the need for blood is still there.
Thank you for reminding all these challenges that concern the blood transfusion. There is also another one, is that the info of the donor. Some donor can have infection, virus or bacteria, and this can be transmitted to the. Of course, in times the blood are tested, they are clean, but in emergency, when you have to bring donors in an emergency field and take the blood, maybe you don't have time to be let's say a very clinical grade. So that's a very important issue. In France, we had a very important scandal past years.
In the 90s, right?
Absolutely.
Yeah. Today, you could say that the blood is relatively safe. The occurrence of transfusion transferred is rare, but still, the product, for example, that we're going to be supplying will be totally pathogen-free because of the standards that we're manufacturing.
Thank you for reminding the challenge that affect the blood transfusion. How can I say, industry? Or let's talk a little bit about yourself and how. Who are you, Dr. Ari? Who are you born, and how? What was your familiar environment? Do you grew up among scientists, or is it something you discovered by yourself lately in high school? Maybe we can talk about a little bit about yourself and how you finally ended into becoming a scientist, and how you become a bio-entrepreneur.
I was born in the United States, but I grew up in Israel since the age of 6, and that was a long time ago. My parents, my mother is an artist. My father taught engineering, industrial engineering, but I had the inclination towards science from a very young age. And one of the first memories I have is from 1969, from the landing on the moon. I remember seeing that in live when I was 4 years old, and how it moved me, and how that opened the craving for exploration. And so that started my beginning as a scientist. But later on, I kind of drifted towards the biology, the love of animals and chemistry, and I was a pretty nerdy kid. I read science fiction books, and I did experiments. And when I used to go out on trips in our youth movement with friends, I would come back with backpacks full of fossils and stones. In high school, and I continued studying biology also after my military service. So that was my direction, and I always had an eye to the biology and to the microscope, and another eye to the sky, to the telescope.
And why did you choose the academic career path? How did the fire of entrepreneurship or being in the industry, because you have a very tough background also in the biotech industry, how did you manage, in your mind, that maybe the academic career was not for you and you wanted to be more on the side?
Yeah. So when I started studying biology, I had a feeling that it's more about running after butterflies with a net, and studying them. It was more of the. But one of the courses that I took was an introduction to biotechnology, and that course, Professor Amichai Freeman at the Tel Aviv University, he gave a course that described what you could do in biotechnology, and actually the understanding I could use microbes and microorganisms and cells to produce products that could save lives, that could remediate the environment, I think that that blew all of my fuses. That really turned me on, and I decided then that I would take my career into biotechnology. And so my master's PhD were in microbiology and biotechnology. In between, I had a year in between them, one of them in which I was participated in the International Space University, and also just in order to bring together my love of space exploration and biology. But in the PhD studies, when I was doing my experiments, I understood that I want to be where things are, where application is, where we'll actually not doing only research, but actually developing products that have impact on humanity. And I decided that I'll go into the industry once I finish my studies. And actually, I was very lucky to have friends that brought me into a startup company at the year 2000 when I finished my PhD. Actually, since then until now, I was in different startup companies in all kinds of areas, from diagnostics to research tools. I was in, if they're biologists, lab people in on this call. So I was in a company that was in Vitrogen and life technologies, and now Thermo Fisher for almost 10 years. I was in a pharmaceutical company developing Parkinson's treatment. But in the end, I always felt that I want to do something of my own. And I was, for many years, I think for all of those 20 years in the industry, I was thinking, what can I do? Here I bring another experience that I had when I was in my bachelor studies. I used to be a paraglider operator. As a sport, I used to fly a paraglider, and I really sucked as a pilot. And I crashed up in the Golan Heights, was severely wounded, double pneumothoraxes, when both of your lungs explode.
Wow.
I lost a lot of blood, and I woke up on a helicopter with a blood transfusion going into me. And that was in 1990 when the HIV was in the rise. And the truth is, I was a bit scared that maybe my blood transfusion wasn't tested properly. And from that experience, and of course, the doctor that treated me, that did the field surgery that saved my life and gave me the blood, said, "Don't worry, the blood was tested, but it still was bugging me. How is it supplied? How is it? How good is the supply?" It was really strange to me that pharmaceutical products are tested so rigorously and have such high standard testing, and here we're taking a body fluid from one person, giving it a few tests, and giving it to the next person. As safe as it been made, it still seemed strange to me. Very not fit for the 21st century. And as I was going by until actually 2012, as I was scouting for different technologies in Israel, I came across a company spun out of the Technion, a company called Excelta. And this company has a technology, growing stem cells in a very special way that allows to grow the stem cells in a unlimited volume, in an unlimited, you could scale them up in an unlimited way. And at a certain point, I understood we could take this technology of cultivating the stem cells and producing red blood cells, and actually solve, if not all, but most of the supply of red blood cells. So from that point, I started calculating and seeing how we could do it, and started RedC. And we're actually in the lab since 2019, working on the process, applying the technology, the IP, the cultivation technology from Excelta for the cultivation of red blood cells. And maybe this is a good time to go and tell you how we're going to do this.
Absolutely. You made a fantastic transition. Thank you so much. Maybe one question on Accelerate. Accelerate is a R&D startup company, and they don't have by their own the will, to go further. They were seeking an entrepreneur, right, to develop further.
Excelta is mostly a company that gives services and for development and licenses their technology. So there's a perfect fit here. We're using their technology for the production of red blood cells. And so there's a license behind that and everything that has to go with that. But the technology is the basis for large-scale production of red blood cells.
Fantastic. Maybe we can now talk about RedC. So you company in before the COVID, right? Just before the COVID.
Yeah. So it takes time to raise the money to do the licensing, but since 2019 we did about a year of proof of concept, and from 2020 we're taking the process that we started developing, working in and slowly and gradually growing the scale, dynamic cultures, and learning the process, how to apply the technology into the production of the red blood cells. And so, to solve the supply, in the end, red blood cells, their body is to be an oxygen carrier. And there were many attempts throughout the past several dozens of years to solve this problem of oxygen carrying capacity for our body. There were several companies that developed chemical oxygen carrying capacities, different ways of encapsulating hemoglobin, polymerizing hemoglobin, other materials like perfluorocarbons. All of these materials have very high shelf life and oxygen carrying capacity, but they turn out to be toxic most of the time. So you could administer them, you could life of a person at that moment, giving them the oxygen capacity, but mostly these materials do not agree with our body and cause indirect effects. So the other direction of producing the oxygen carrying capacity is by manufacturing actual red blood cells. And to manufacture the actual red blood cells, it's using stem cells. And this is what we're also doing. But there are stem cells. The technology for using stem cells has different aspects of it. So many of us know that perhaps 20 years ago, and even up to about a decade ago, embryonic stem cells, or stem cells that were sourced from embryos, were very easy to get, but there was a lot of ethical controversies about the use of them. And fortunately, they were set aside because of a very important discovery that a professor from Kyoto, from Japan, made called Yamanaka, where he developed a way of neutralizing a few genes and producing a type of stem cells which are called induced pluripotent stem cells. These stem cells are stem cells that the source of the cells can be any cell in a body. From adult people, for example, you could use skin or red or white cells from a human sample. You could take them from a person, from a patient, and in order to produce his own cells, or you could take them from a person that has a special characteristic and produce a cell line that will produce a target product. So stem cells are cells in our body. They replenish all of our tissue and produce more blood, more skin cells. But when we look at them at an industrial level, they have two very important characteristics. The first is that the population of stem cells replenishes itself, and you could produce unlimited amounts of stem cells if you know how to do that. The second important part of characteristic of stem cells is that if you know the chemical and biological process to turn these cells into the target cell, so you can produce any cell that you theoretically, any cell that we have in our body. And this is at the essence of what we're doing. So we're taking the stem cells, the pluripotent stem cells are stem cells that are from a donor with characteristics that we know, and we're able, and this is special about our technology, is that we're able to grow these cells in suspension as single cells, unlimited, which means that the potential to scale up and grow these cells in a very large bioreactor is unlimited. And of course, and this is the process that we're developing, is taking these stem cells and differentiating them into red blood cells. Mm-hmm. Many groups know how to take progenitor cells and cells that have been taken from our body, from our bloodstream, and relatively easy, nothing's easy, but are able to take these progenitor cells and turn them into safe and effective red blood cells, but the amount that they're able to produce is limited because the cell is already committed to be a progenitor cell. The number of multiplications that it could do is limited, so the end point that you have is relatively limited. We're circumventing that challenge by the fact that we're growing very large amounts of naive stem cells when these cells are still differentiated, what's called undifferentiated cells. And when we have a very high mass of cells, only then we start pushing them from the state of naive cells to the progenitor cells and to the red blood cells. So this will allow us to finish up with a higher amount of cells. And because the cells are grown in suspension and in a bioreactor, the efficiency and the density of cells is much higher, meaning that the cost per cell or per volume unit can be lower. And also, as we could see all around us, this field of cultivated meat and the technologies for very high, large-scale cultivation of stem cells and cells to produce a product. These technologies are growing as we are speaking all around us. So the platforms, the materials, and all of the industry that brings all of the growth factors and raw materials that are needed is also growing, meaning that the cost of raw materials will go lower and lower. So our challenges are, on one side, making a very highly efficient process of differentiating, of growing high mass of naive stem cells, differentiating them into red blood cells, and of course, making them as low cost as possible, meaning to product that could compete in its cost to the cost of a donated blood unit. So maybe a bit about the numbers here, which we didn't talk about before. A blood unit today in income countries is about $200 US, more or less. Some places they're more efficient. So the amplitude could go from $100 to about $400 for a processed red blood unit when a hospital will purchase it. But on that, there are additional costs, the cross-linking, different cleaning and separation and processing of the red blood unit. So in the end, we have to compete with the cost, the minimum cost that we have to compete with is $200 per unit. And our target at the end of the line is to produce a blood unit at the cost of $50 per unit. This will not happen immediately. One of the main points is economy of scale. In order to reach such costs, you have to have very large scale. You have to be able to produce. You have to have the purchasing capacity to buy low-cost raw materials, and you have to have the production capacity of large amounts of cells so that the cost per unit will go down. So we'll achieve this in time. It will take several years. And when going to market, maybe we'll talk about that later. Going to market will be gradual as our capacity grows.
Fantastic. Just let me remind the source of your cells. It's induced pluripotent stem cells, right, from one universal donor.
Right. Yeah. Maybe that's explained here. So this is another section and a very important section of our work, and that's how do we produce universal red blood cells? How do we produce O-negative cells, and perhaps even more pristine and universal cells than only O-negative? Red blood cells are very interesting cells. They, at a certain point when the cell differentiates, these cells eject. It's called enucleation. These cells eject their nucleus, meaning that a red blood cell, when it's mature, is not able to multiply. It will not get additional blood cells. This is also the reason why the red blood cell has that very special concave structure. It's the red blood cell is red because of hemoglobin. Hemoglobin is an iron-carrying molecule that has the capacity of chelating or holding an oxygen atom molecule when it goes. But the structure allows also very high surface that allows the red blood cell to absorb the oxygen very quickly and give off carbon dioxide in our lungs. But the concave shape that gives the cells deformability and flexibility that allows them to bend and stretch and go into the smallest blood vessels that we have. That's on one side. These cells are also very small. They're about half the size of our standard red blood cells. They're 7 to 8 microns in diameter. Also because they do not have the nucleus. But this means also that these cells have a very high density. You could separate them very easily, and because they're small, you could filter them very easily. A very important asset for us in order to make sure that we have no remnants or residual white blood cells or other cells that have not differentiated in a blood transfusion that we get. And how do we get the universal cells? So a red blood cell has about on red blood cells, we have about 35 different antigens. Among them, there's a variety of about 350 variants in humanity, meaning there's a lot of complexity here. But the main ones that contribute to rejection of blood and the adverse reactions of a red blood cell are most important the ABO group. You know, a person who has B cannot donate to A, and vice versa. O can give everybody. And you have the RHD group, which contributes to the positive and negative group. And of course, negative is the more universal. So an O-negative blood type is the best. There are additional antigens that are in the human population around the world. And we're bioinformatics study, and interviewed many hematologists in order to understand what a truly universal blood type will be. And what we'll be doing, and this is a project we're going into in the next year, we're going to be contacting some of these people. It's a very rare blood group. These really universal people are very rare. They're about less than about 1 out of 10,000 in our population. And these people can donate to everyone and cannot accept from anyone but themselves. Believe it or not, these people have their own blood stored in case they have to go to an operation, or if they have an accident, they have their own blood stored for them, but they could give everyone. And and people that are heavily transfused, like people suffering from sickle cell disease or thalassemia, can receive their blood and will have a minimum side effects from these people's blood. We will be working with several of these people in order to produce, and this is a one-time donation that these people give. We will be taking a blood sample for them, producing cell lines from their blood, and selecting what we will call the mother or the father of all blood donors, and making a blood bank, a master cell bank that of a cell line that this blood type will fit all humanity and will be able to produce blood for all humans forever. This is one of the assets that we're building that will be able to be a raw material for our process for the production of universal red blood cells. The technology that enables us is that Yamanaka technology coming from the Kyoto University.
So to better understand the logistic of your business, so you will provide this iPS cell derived from these universal donors to the local hospital that you are going to collaborate with them. You know how this is, and the iPS cell produced in-house by you will be to the different blood agency and hospital and clinics, etc. Right? And they will organization themselves.
So our vision, we don't believe that hospitals or perhaps even the blood agencies themselves will have the capacity to produce the red blood cells themselves. But the way we vision it is that we will have production sites, either with collaboration companies or with global bioproduction companies that will establish production sites all around the world. Like in every continent, in every region, you will have 1 or 2 blood production plants that will use, as you said, the stem cells that we will produce and the process and the raw materials. Each one of these production sites will be under the local regulatory system, because every region has its own, every country has its own regulatory process requirements for the production of pharmaceuticals and especially blood. So we will have to meet the requirements in every place. And also another point is for logistic reasons, we want to have to diversify the supply chain all over the world. You want to make sure that that the raw materials can be brought from different places, but also the production will be near the hospitals. So we believe our vision is that we'll have multiple production sites in every continent that will produce the red blood cells and be able to supply. And another part of it is that we will have redundancy between the regions. If one region has a catastrophe or there's an earthquake, people are famine in one area, people cannot donate in one area because the production will be the same, uniform, and it will virtually be the same product. We will be able to move blood from one place to another. Today, blood is a national treasure. Countries do not share blood readily. And in Western countries, we know that, for example, that for a short period there was a supply of blood to Ukraine. When there was the earthquake in Nepal, there was some blood supply. But usually this does not happen. And each country is at its own. This way, when it's an off-the-shelf pharmaceutical product, we'll be able to save people everywhere. And also, when the capacity will grow, we'll be able to send blood also to low-income countries. Many countries in sub-Saharan Africa, where the occurrence of sickle cell disease is very high, where the amount of people that suffer from tropical diseases that cause anemia in the thalassemia belt that we have coming from the Mediterranean all the way to Southeast Asia. We have extremely high occurrence of anemic thalassemia patients. All of these places, we have very large numbers of anemic and transfusion-dependent people that require blood. And very unfortunately, these are also, in many cases, countries that do not have the capacity to supply blood properly. So, and see a lot of countries that could supply maybe 25 to 30% the amount of blood that they need. If we'll be able to grow in capacity and source low-cost raw materials, we'll be able to produce blood that will be able to be supplied to everywhere, anywhere, really saving people.
Your solution is so elegant, so powerful, and so what can I say, so efficient. It's the whole process from the iPS cells to the red cells is protected by 50 patent, right?
Right. The technology that we're using is for the first scale up of the naive cells. The process of taking stem cells and differentiating them from the point that you have progenitor cells is very widely known, and there aren't a lot of variations to that. We have a lot of know-how that we have learned in the process of development, a lot of tricks, but not a lot of things that could be defended by patent. But the actual technology that allows us to grow the iPS as single cells in suspension and reach extremely high densities, that technology is very highly patented. As you said, over 50 patents, 7 patent families, and that is the basis for the.
And what is the yield of red cells obtention? Do you have a 100% red cells when you do the process, or do you have to filter white cells?
Any process like this is not 100% yield, and when we have the cost by raw materials on one side, making an efficient and short production process is the main biological or the main engineering process, the challenge. So here, this is what we're doing: how to make the best out of every step, how taking in our differentiating process, making it as efficient as possible, making it as short, and reaching the highest yield. And this is something that we're growing in the ability, from a process that took over 40 days, we're going to get near to about a month at the end of the process. From yields that when we started were at the 30 to 40%, we're taking them up to the 80 and 90%, and 90% at the target. There will always be cells that have not differentiated well enough, that have not gone into the direction that you want, and that will have to be separated. So that's part of the cost, that 10% cells and debris that go into the product.
So as you are raise funds right now, maybe we can discuss a little bit about the business model and how your vision about how you are going to make money. It's of course biotech with the code, but it's a company, so we have to this point.
Of
How
course.
do you expect to make money? What would be your business model?
Yeah. So RedC Biotech, we envision ourselves as supplying the core technology, both out of licensing. So when we will have production sites all over the world, and we will supply the stem cells and the basic materials and the know-how. So we'll have licensing fees, and the companies that will produce will be on the red blood cells, so units. That's our main model. This will enable us to continue and develop additional cell lines, will enable us to perhaps look into other directions. Because when looking far into the future, the best solution will be to have a whole blood transfusion, a blood that consists not only of the red blood cells, but also from platelets and from an artificial plasma. So if we take artificially produced platelets, by the way, there's a company, a group in Japan that are already doing clinical trials of iPS produced platelets in Japan. If we take all of these components, we'll be able to produce an even higher, for let's say, for have anemia, all they need is red blood cells. But people that go into operations or suffer from an accident and different trauma treatments, women in labor that lost blood in newborn babies, these people need both platelets, plasma, and red blood cells. And in the end, we would like to supply the red blood cells are a big enough challenge for now, and a very big challenge as it is. Right now, we're scaling up. We're at the very first stages of taking our process into dynamic, into bioreactors, and it will take us about two more years to reach preclinical testing, meaning that it will take us at least 5 more years to reach after we've done some clinical the required clinical testing. So we're seeking investments as we are speaking, and be happy for people to contact me and talk to me after that about investments.
Can we talk about the amount you are seeking, or do you prefer to keep it?
So I would like to, that's something we're at this point right now we're seeking $5 million, but of course the requirements will grow as the capacity grows, as the scale grows, as we go into preclinical and clinical testing. All of these, the amount of it is required grows, can't say exponentially, but grows considerably. But at this point, that's what we're seeking in order to make us, to bring us to a point where we could produce a scale and quality that could go into preclinical testing.
Great. So let me launch out for you. So if there are investors here who would like to support such an amazing project, so you contact Dr. Ari Gargir directly to talk further about this opportunity. Thank you very much, Dr. Gargir. I think we have a very great view of what you are doing. And maybe to conclude and to discuss something lighter, maybe we can talk about other possible, maybe science fiction application of your solution. There are a lot of hype nowadays about rejuvenation and longevity. Could we imagine maybe mixing your universal red cells with growth factors to rejuvenate people? Is it something that is possible, or even maybe used by sportsmen illegally to clean their blood, be more performant?
Right. We all know the story of Lance Armstrong taking erythropoietin, epogen. That's one of our main raw materials. That's the main molecule. Commands a progenitor cell to turn into a red blood cell. So that's the famous story. And there, as you said, there are many stories about taking blood from young people and transfusing what people like to call vampire rejuvenation. Red blood cells that we're going to be producing will be very high quality, and perhaps most likely will have a longer shelf life, because in our body we have cells that are between 1 and 120 days old. The cells that we'll be producing are all harvested at once, so the cells will be very young and high quality, and perhaps survive in our body extended time. Some preliminary experiments show that is very. I have no idea what effect the red blood cells per se will have on the rejuvenation of a person. Also, most likely, the growth factors and the environment of the blood that comes with the red blood cells is the one that contributes most to rejuvenation and to the longevity. But who knows? That can be tested.
I have also two other questions about other possible market. What about the veterinary medicine market? Is there a need of blood transfusion for our pets or for pets? Or I don't know, because I don't know anything about this market, but maybe you have some key for us.
Yeah, so our pets, because they're mammals, and like we are, they need blood, and dogs, and also cats occasionally get blood transfusions. The veterinarians, in many cases, have to struggle in order to get blood transfusions. There, in some places, there are blood banks, and some veterinarians take upon themselves to produce blood banks to supply blood. But in the case of at least the pets and the animals that I studied, meaning cats, dogs, and horses, it turns out to be that their blood types are more complex than ours. As humans, we're relatively lucky that we have a relatively system that enables us to share blood easily. When it comes to our hairy loved ones, it's more complex. And then, of course, seeing a veterinarian.
We have a freezer, but
there's a universal is the market.
The market is maybe difficult.
The other point is the size of the market.
It's maybe difficult to evaluate.
So
but there is a need.
There is a need, yeah. And perhaps absolutely. The next stage will be pet.
Absolutely. Maybe a last question about the. We can also imagine that the red cell can be also be a drug carrier, because they are for the vaccine. There are a lot of research that try to develop phospholipids, to envelop the mRNA or any kind of drugs. They are good, but they are not perfect. But now you have real cells, with you that can be a drug carrier. Have you thought about that?
Right. Yes, and when we talked in the beginning, or perhaps I forgot to mention it, but another application other than transfusion is advanced therapies, and there are quite a few companies around the world that are developing the use of red blood cells for advanced therapies, and this goes into two directions. There used to be a company called Rubius, and there is a company now called Scarlet Therapeutics in the UK that produce red blood cells that express on their surface different antigens, which could elicit an immune response and against cancer, or as immunization, or perhaps very similar to a CAR T kind of a treatment that is used for cancer treatments. So one of the most important things about red blood cells is because if they're universal or if they're O negative, they are not rejected by our body. The other important point is that these cells can survive in our bloodstream for up to 100, 120 days, which means that you have an agent that could be swimming around in our bloodstream for over 3 months. And so we mentioned those companies that coat or express antigens on the surface of the cells. And there are another 6 or 7 companies around the world that are developing different methods of pushing different therapeutics into the red blood cells, either by using electrical shocks or osmotic shocks, but they're able to push different therapeutics into the volume of the red blood cell, using the red blood cell as a carrier. And because the cells will be metabolized or dismantled in our bodies slowly and gradually, or even if you could direct them to different targets, organs. So it's an ideal carrier for these therapeutics. Perhaps maybe a longer. There are a few clinical trials already out there using blood from blood donations, but it's a different market. It's a very promising market, and opposed to blood supply for transfusions, where the gross margin is smaller. Over here, these are very advanced therapies that have very high margins per treatment.
Fantastic. This is very promising and exciting. You are on a very important domain and very impactful. Thank you so much for being my guest. Maybe if you want to say a last word, maybe did I question that you would like me to ask and I forget?
No, I have a feeling that we covered it all. Maybe just to summarize, that we're talking about a very great or a very big unmet need. People around the world require blood and will require blood. This is a market that will not diminish. It will perhaps only grow. It's an extremely large market with a very large potential that has not, which we're maybe talking about today, about it maybe half of the supply around the world. So we still have an enormous way to grow and supply. And most important, it's a global humanitarian need. And we're going to be saving lives of literally of millions of people. And I think what we're going through now in Israel and many incidents around the world, we know that it's extremely important. We know that, but based on this, I think just let's hope for peace and prosperity for everybody. And I really thank you, Ari and Harold, and the whole, all of the people that were listening for joining. And I'm very happy to tell RedC's story here.